Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. There are many reasons for doing research on the brain, for example, with the tools of computational neuroscience. Curiosity is one reason, possibly help to build better AI is another. A third is to help cure brain diseases or help people with disabilities, that is clinical neuroscience. The latter is the topic of the day. That is computational neuroscience for the clinic, or computational neurotechnology, if that is a word. How can computational neuroscience contribute to develop neurotechnology? That is to help people with brain disorders and disabilities. When it comes to clinical interventions, there are several options, including talk and behavioral therapy, neurosurgery, treatment with drugs, and use of electric and magnetic fields to record and stimulate nervous tissue. All techniques can be aided by computational neuroscience. It seems to me that our field's main contributions to clinical treatment so far has been to assist in development of electrical and magnetic recordings and stimulation. Interestingly, Scribonius Largus, living 2000 years ago in Rome, working for the Emperor Claudius, described the therapeutic application of the live torpedo fish to relieve pain from headache. Like the electric eel, the torpedo fish can generate electric shocks, and Largus treated the headache by placing the fish on the head where it hurt. Anyway, the topic of today's podcast is the use of electric recordings and stimulation to help patients, and in particular the role of computational neuroscience in facilitating this. The typical format of the podcast is me talking to an expert, but this podcast is different. It was recorded live with a panel in front of a live audience, as they say, as a part of the conference program of CNS 25, the 34th annual Computational Neuroscience meeting, which was held in Florence, Italy, in July this year. Thanks a lot to the organizer of the conference for allowing me to do this. For me, it was particularly exciting that this Computational Neuroscience meeting was arranged about 2 kilometers from the burial place of Galileo Galilei. He is famous for many things, also for saying natural laws are written in the language of mathematics. This is like a credo for computational neuroscience, and of course also for many other theoretical science disciplines. I invited 3 panelists, all with lots of experience in neurotechnology and computational neuroscience. The first is Anthony Burkitt from the University of Melbourne in Australia. He has, among other things, worked in what is called medical bionics. That is to make neural prostheses to help deaf people to hear and blind people to see. His work has included the development of retinal implants, a bionic eye, and cochlear implants, a bionic ear. The second is Nala Yousif from the University of Hertfordshire in England. She has done a lot of work to understand the mechanisms of therapeutic neurostimulation, including deep brain stimulation, nerve stimulation, and magnetic resonance-guided focused ultrasound. Here, the main aim has been to optimize these treatments for movement disorders such as Parkinson's disease and essential tremor, that is, involuntary rhythmic shaking of limbs. The third is Esra Neufeld from the ETS Foundation and ETH in Zurich, Switzerland. He has worked a lot on understanding the interaction between neurons and imposed electric fields, simulation tools for medical device safety, and various non-invasive neuromodulation techniques. In our 1-hour session, we discussed many aspects of working on neurotechnologies, not only the technology itself, but also the challenges of translating scientific findings into useful products and therapies that can help patients, and also trying to commercialize these technologies. We also discussed the sociological differences of working in a more clinical setting with medical doctors and patients, rather than in a more traditional academic setting. And of course, we speculate about what the future might bring in terms of new technologies for use in the clinic. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking. For example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast where you also can watch the videos. I hope that many of you can sign up to become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners and hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Note that there is no video recording of the present podcast. Okay, here is our panel discussion. We enter immediately after I have introduced the topic of the podcast to the conference audience. So, then now to the podcast itself. I mean, one of the greatest successes, I think, when it comes to like neuroprosthesis is actually these cochlear implants. And first time I visited Australia a few years ago, I came across this book at the airport about a person called Graham Clark, who is obviously should be a household name for all of us, because ibuprofen. Oh, sorry. Oh, so drugs are also useful, not only neurotechnology. So anyway, this Graham Clark, I read this is a quite moving story. He grew up in a sort of like town, a little bit in rural Australia, as I understand it. His father was a pharmacist and he was deaf. And when they asked him what he wanted to do when he got older, he said, "I want to be a doctor that fixes ears." And that's actually what he did. He made cochlear implants. So this is also, I think, how you, Tony, you got into this like neuroprosthesis and, well, neurotechnology world. So maybe you can tell a bit about that.
Certainly. Can people hear okay? Good. So I was very fortunate to work with Professor Graham Clark for 10 years, what used to be called the Bionic Ear Institute and is now called the Bionics Institute, which is where the first multi-channel cochlear implant was developed. But my pathway into computational neuroscience really began following my PhD in the group of Peter Higgs at the University of Edinburgh, long before the Higgs boson was ever discovered. And then through statistical mechanics and Hopfield networks, which of course many people here probably had a similar sort of path into computational neuroscience. And it was really that sort of grabbed me. The idea that we could actually use the methods that we had developed, the mathematics, the computational methods that we developed in physics, to help understand how the brain worked. And it's Professor Clark who really gave me the first opportunity to work in this area, who wanted people who would have an understanding of the mathematical computational techniques to be able to apply those to understand how the nervous system worked. And my colleague David Graydon, who's also here this evening, started at about the same time with me at the Bionic Ear Institute. So trying to understand particularly how the auditory pathway worked. Now it'll come as a bit of a surprise to many of you to know that the original cochlear implant really just did stimulation. So it was what we would call an open loop sort of technique, where you would just sort of stimulate, you'd actually get the patient to explain what it was they heard, and so you would adjust the levels up and down and do all sorts of decomposition of the frequencies using Fourier techniques, and then stimulate the appropriate nerves. So what Graham really wanted to do originally was to really get a better understanding of how the auditory system is encoding speech for normal hearing, and then to try and mimic that as closely as possible with the cochlear implant. Now, that's actually been incredibly successful. There's well over 1 million people now worldwide who have been implanted with the cochlear implant and essentially leading fairly normal lives. Children who get it early enough can actually go to regular schools that don't require specialised schooling and really lead totally integrated lives. So I think it's been transformative in terms of the patients who have received this implant. I was there for 10 years and then moved to the University of Melbourne, where we were developing and applying the same techniques to retinal implants. So it's taken a long time from the development of the cochlear implants to really develop the retinal implant, partly because the complexity of how the eye works. And of course, your eye is constantly moving. It's like almost like trying to throw a transistor into a washing machine and get it to work. It's how it's sort of described in very rough terms, because the eye has so many neurons in it. It's such a complex processor that actually does a lot of processing within the retina itself. So it's taken a long time to develop the technology, miniaturize that, and make it safe enough to actually put it into the eye. So we were amongst one of the groups in the world who were developing these visual prostheses. There are a number of different approaches that are still being pursued around the world. Ours involved a subretinal implant that sort of goes between the different layers of the nerve cells in the retina, with the idea that you would stimulate the surviving nerve cells there that could then send the signal back to the vision processing centers of the brain to be interpreted as vision. Now, the next sort of like really big challenge there, and this is where computational neuroscience really plays a big role, is to be able to develop stimulation strategies that aren't that sort of open loop, but that it can actually measure the stimulation that you're generating in the nervous system, in our case in the retina, to be able to then titrate the amount of stimulation that you do according to the level of stimulation that's being evoked by the stimulus itself. So that's where we get a sort of closed loop type of algorithm, which I think really represents the next generation of these techniques. And in order to do that, we really need to understand how we can model and get an accurate representation of the amount of stimulus that will be generated in order to sort of close that loop. And we're now using that technique also for brain-machine interfaces, and hopefully that will also be next generation type of use of this technology for people with all sorts of disorders, from paralysis, anyone who's epilepsy, Parkinson's disease, there are a whole range of sort of neurological conditions where these sort of techniques can be produced. But perhaps that gives you at least a flavor of what
absolutely.
I do.
I mean, so the key thing here is that you have some kind, I guess with the visual prosthesis, have some kind of camera that takes an image, and this is converted into like electric currents that stimulate the retinal cells. And so also for you, Nala, right, you also work with electrical stimulation, but then in the context of deep brain stimulations for Parkinson patients and maybe other people with movement disorders.
That's right. Yeah.
So Nala, yeah?
Yeah, so that's right. So about 20 years ago, I did my first postdoc, and I joined a clinical group actually who were, and still are actually, doing deep brain stimulation. So most people here will probably know quite a bit about deep brain stimulation or something about deep brain stimulation. So, I mean, essentially, prior to deep brain stimulation, for things like Parkinson's disease and tremor, the typical treatment was actually lesioning specific targets in the brain. So, for example, for tremor, it was usually a nucleus in the thalamus. And it was actually in the late 80s when a surgeon who was performing such a thalamotomy just decided to trial high-frequency stimulation rather than the low-frequency they typically used for kind of mapping out the area that they were going to lesion, and found that the high-frequency stimulation actually suppressed the tremor. And so that was really the start of deep brain stimulation. And following that, you got basically the development of chronic implantation of electrodes into disorder-specific targets. So for Parkinson's today, the targets are typically subthalamic nucleus and the globus pallidus. And then for essential tremor, it's usually still the thalamus, the ventral intermediate nucleus. And then the group that I've worked with, so starting from that first postdoc, but it's been a long-running collaboration to today. They also implant patients who have dystonia. So essentially, for the first 20 years since DBS became kind of an approved treatment, which was more like the late 90s, the technology didn't change very much. It was open loop, as you've described. So a current was injected into the brain, and they could tweak some of the parameters, like the amplitude and the frequency and the pulse width of the stimulus. And the clinician would just basically have a look and see if things like the tremor was suppressed, if the rigidity was improved, and adjust parameters by trial and error. And for about 2 decades, that didn't change very much. But over that period, obviously, computational modeling was one of the things that helped to change the hardware and the software. And so lots of different kinds of modeling.
Yeah, but maybe because I guess there are like two elements. One is that you have to model how the currents actually sort of sets up electric fields in the brain tissue, and the other part is how that electric field affects the neurons.
Yeah, exactly. So a lot of the modeling, can be, you can kind of almost split it into those two. So typically, initially people would just look at kind of point sources for the electrical stimulus and look at how that spread in tissue. And then that soon developed into more engineering type models like finite element models, where you can represent the geometry of the electrode more accurately, and you can also represent the tissue properties with different levels of complexity. So you can assume it's just homogeneous and it conducts the same in all directions. Or you can go as far as get your values for things like electrical conductivity based on medical imaging. And then, you have kind of very fine level detail of how the current spreads. And then what people often do, myself included, is take those estimations of the electric field from those models and apply them as an extracellular potential to neuron models, or more typically axon models. So axons, as many of you will know, are more easily stimulated or excited by electrical stimulation, and so that's become really a focus for research, actually. And it's one of the ways that people are moving towards closed-loop systems, actually, is to kind of use these models to estimate the spread of current and trying to constrain your parameter space in terms of the actual clinical system based on those models. Yeah.
Great. So Esra, so you worked on many aspects of, I think, neurotechnology, and one of the things, maybe start with that, is like you also help in some like spinal cord stimulation to help to aid people to learn or to help people walk.
Yes. So maybe I take a bit more of a historical perspective. As you said, we originate from the Swiss Federal Institute of Technology, the ETH, but we were basically a group in the field of EM exposure safety, when mobile technologies were considered a questionable thing, and where there was a need to clarify scientifically how to test, how to assess the impact on things from brain activity to heating, tumor risks, and so on. And so it was decided to create this non-profit research foundation to act as a bit of a bridge between regulators, industry, basic research, and later on healthcare providers, focusing on questions of safety to telecommunication, MRI safety, and so on. And my first touch with neurons was actually in that time when we were looking at MRI gradient coil inducing unwanted peripheral nervous system stimulation. That's the first time we did the kind of coupling that you described with finite element methods and axonal dynamics. And so over the years, we strongly shifted our focus to the beneficial applications of various physics, EM, but also ultrasound, thermal, others, interacting within the complexity of the anatomy with biophysical and physiological responses. And so in the context of spinal cord, as you were bringing that example, this started off with the question of neural interfaces to nerves. How can you build invasive implants that target certain fibers, certain fascicle or subfascicular components to directly activate muscles to restore locomotion in desired form? Worked nicely on rodents, did not work nicely on humans for reasons which we now understand, mostly thanks to modeling. But so the next step then was the aspect of modulating existing motion patterns within the spinal cord and triggering those patterns. That's work done with people like Silvestro Micera, EPFL Pisa, or Grégoire Courtine. And we were responsible for the modeling side of things, where we again take these personalized images that you described and create models that integrate imaging information about anatomy, about tissue properties, about connectivity, about functional responses to create personalized models. And I would say that's a typical example for that aspect. And the outcome was that this process of tuning, which in the case of spinal cord stimulation could take months, was in the end reduced to a single day, and that within that day, people regained the ability of walking, cycling, swimming motions with weight support and subsequent physical therapy. But the models brought it to the point where you could right away identify a personalized program that worked, that got people to walk.
So, I mean, I think like when the cochlear implants, that's like more than 1 million. And I think I read somewhere that like deep brain stimulation, where they then surgically put this sort of like deep electrode into place, that's like 160,000 people or something already. Then, but this like spinal cord stimulation, how common is? I mean, is that
the paraplegic, or now we're working on tetraplegic, is certainly less common, which is why when they founded the company, the financial investors redirected it first to blood pressure regulation, which you can do with the same technology. Yeah. Much less impressive, much less dramatic, but far wider population, of course. And I'm glad to see that the investors, now that they have these first success applications, that they are not giving up on those rarer cases, because it's also a matter about what choice you have. And for the paraplegics, there is very little alternatives around there. So I'm happy to see that it's getting back to that original driver of the research.
Because expenses is obviously one important thing. And I think you, Tony, you worked on some other kind of brain sensing device, right?
Yes, that's right. I mean, one of the really interesting developments has been the development of what we call the stentrode, which is a stent electrode. So just as stents go into the heart, they can also be put into blood vessels in the brain.
So what is meant by stent electrode? Is just the name, or is
it is literally a stent is used in the heart to hold open the blood vessel, but embedded in that are the electrodes. And that the electrodes, each individual electrode then actually has a wire that goes back to typically a unit that's actually implanted in the chest, so totally implanted. But by putting the electrodes, the stent array into the brain, you can target the motor areas that are responsible for movement. And so in the first instance, you can actually just measure the activity of those motor areas and use that as a way of being able to interact, then for the patient to be able to interact with a computer. So for patients who are completely paraplegic, quadriplegic, or who have things like motor neuron disease, this can be transformative in their ability to be able to communicate with people outside. Ultimately, what we'd also like to be able to do is to use those electrodes to stimulate and perhaps to then control things like limbs and so forth.
So because that's what you mentioned this with closed loop. I think several of you mentioned that closed loop, meaning that you have, well, ideally maybe the one device that both records and stimulates, and that you can do this on a. So that is maybe like a key development.
It is. The reason for that is that it doesn't require brain surgery. So a lot of the other ones, you actually have to take a bit of skull off and put the electrodes in and so forth. Whereas, of course, this can go in an endovascular approach, which is done quite routinely now by the neurosurgeons.
What do you mean? You go inside the blood vessel?
That's right. So it actually typically goes through the blood vessel here that can then be threaded through up into the appropriate area through the blood vessels into the cortex.
Yeah.
If I may comment on the aspect of control, I think it's important to point out the role of modeling in control, because modeling plays a role in deciphering or in maximizing the information content and in extracting the content from the signals. As you know, you're an expert on neural signals. Those signals have few dimensions of information, but you have ability by playing with how you record them to extract the information that you care about with maximum ability. And modeling can help with that. Then you are working in a huge dimensional parameter space, but the underlying functionality you're trying to control might have a lower dimensional manifold embedded in this high dimensional space. And so if you create the model of the responses and the response to perturbation by stimulation, that also means that your model can become part of the device. So the model becomes the logic by which the device decides the spatiotemporal activity, how the source term that it applies to the system. And then the modeling aspect also then affects the design of the neural interface for stimulation, because you need to know how and where to stimulate to have the necessary selectivity, which you can again do with the modeling. And then you have the questions of safety and regulatory approval, where the model again comes in, translation. There's a whole series of aspects in that work, but specifically for the control, you have the stimulation, sensing, and the model predictive aspect.
I think
so I can maybe bring an example from the work of Antonino Cassara, who is probably in the audience. So with him, we were working on a bioelectronic medicine, an electroceutical implant. And that's a device from a certain company. They did the experiments on pigs and wanted to move to humans. And first of all, the diameter of the targeted neurovascular bundle is about 3 times larger. They need a new implant. The distribution of the fascicles is completely different, the functional aspects. And the question came up with, what should we expect? How will the required currents change? Can we achieve the same selectivity? What device design and what failure mode should we expect? And the work that Antonino did there is he used modeling to reproduce the experiments to get trust in that kind of interaction modeling, compared that to experimental data from the pigs, but then used it to predict how things change when you go to humans. And on the basis of that, the design was chosen, the design also of the stimulator and the currents that it needs to be able to sustain, and the human study was started confirming the results predicted from modeling. So modeling was the basis for translation from animals to humans.
So I think both, Nada and Esra, you also worked on, because if you want to these optical techniques, which are very useful in to study mice and animals, is not really applicable to humans so much, right? I guess you can use infrared, but that's much more diffuse. So it's really either electricity or magnetism or sound. So I saw that you're both working with ultrasound, focused ultrasound. What is that all about? Why do you use that?
Well, I think we work on slightly different types of ultrasound. So with the clinical group that I started working on deep brain stimulation with, the same surgeon also does HIFU, or high intensity focused ultrasound. And this is, again, it's an actual ablation technique. So it uses this ultrasound applied non-invasively, but under MR guidance, to basically destroy tissue in the brain, in the thalamus, to alleviate the symptoms of essential tremor, basically. So, I'm still working with that group on some modeling on that, mainly till now, looking at where they are in the brain and what the impact of the heating is having in that very small region.
So what kind of, because I don't think I've seen any modeling like this on the sounding. There's a lot on this, just not at the meetings I go to. Or the
no, there's a lot of it, and it's a
because then it's like the sound propagation. It's like something that oscillates, like it's different.
You can simulate it across from shear waves and biomechanical simulation as kind of with the nonlinear effect and the image-based, Hounsfield unit-based maps all the way to simplified linear pressure wave or spectrographic methods. But in fact, we also started with that historically in the field of chronic pain, ablation, stereotactic ablation with high-intensity focused ultrasound. Later on, that shifted to cancer, was not successful in cancer, but worked very well for pain therapy. And then do lower intensity and reversible blood-brain barrier opening and drug delivery, where you can use the focused ultrasound to act on the tight junctions and reversibly open the blood barrier and get microcarriers, liposomes and things like that, crack them open the blood-brain barrier, let them through. And nowadays, mostly the low-intensity focused ultrasound for neuromodulation.
That's work that we're doing as well. We're using that low-intensity ultrasound to focus targets for stimulation in the thalamus and so forth. So it becomes an alternative then for electrical stimulation for deep brain stimulation.
In terms of focality, it's much easier to get a precise focus than an electromagnetic in a non-invasive form. Of course, with the DBS electrodes, it's another story, but if you want to be non-invasive, you can achieve centimeter scale with techniques such as temporal interference. With focused ultrasound, you can get a grain of rice, roughly.
Because I guess if you want to inject currents from the outside of the brain, there are limits to how much, I guess, 3-4 milliamps or something is sort of like...
No, when you go to higher frequencies, you can go higher. These sensations and pain at the scalp level, they are strongly frequency dependent. But at typical frequencies, yes, 2 to 5 milliamps.
Yeah. What about like this? So is the ultrasound is shaking? You get sort of like...
The ultrasound has its equivalent in the sense of heating near the cortical bone. So there you have an impedance mismatch, an acoustic impedance, which can lead to heating. But the challenge is rather in the field of low-intensity focused ultrasound. I'm not talking about HIFU, but low-intensity focused ultrasound is the question of the consistency and predictability of the results. EM fields are rather predictable, reproducible, and clear. Low-intensity focused ultrasound, there's progress, but it's still more on the statistical level, and also the interaction mechanisms are less evident. There are at least 5 candidates, and I believe multiple of those are relevant at different regimes. But so the certainty at which you can activate the response with focused ultrasound is not as good as with EM.
Is that because sort of like the mechanical properties of the various types of brain tissue is more different than it's...
I think it might be about, so one of the hypotheses that I believe plays a role, though not the only one, are sonophores, which is in the bilipid membrane constrained by transmembrane protein. You get a kind of cavitation of an oscillation with gas transfer that changes the capacitance, and that combined with the nonlinearity of the ion channels means that you have a charge build-up that ultimately leads to spike initiation. And this is a stochastic phenomenon. The presence of these sonophores, there's just not enough of them. Then there is also the aspect of channels. You have probably another pathway, which is a mechanosensitive ion channels. Here again, not every neuron has them. Their prevalence, and there seems to be also a kind of fatigue effect. The fact is, it is right now less robust and predictable, but it has this beauty of the focality. Another drawback is the fact that in the clinic, to get that kind of focality, you usually have a helmet with about 1,000 transducers that you fix stereotactically, and where you have maybe an MRI, even in the case of HIFU, to make sure that you're targeting the right location correctly. So in terms of the necessary complexity, expertise, environment, and costs, it cannot rival things like transcranial electrical stimulation.
Except that there is one exception to that, because we're developing an endovascular approach that uses ultrasound. So it's using the same sort of endovascular system.
Also neural dust. That's another technology that's being explored. Or if you
it's great that you talk to each other, but you should talk into the microphone at the same time.
Yes.
Good.
So there are ways when you have access to it in a certain way. I mean, there's always the question of what you call non-invasive. So you are bringing it close. It's minimally invasive. Somebody told me once that focused ultrasound is also invasive, and that in fact the cinema is invasive and can cause epileptic fits if you have the right light phenomena. And so where you draw the barrier between non-invasive and invasive. But what I mean is, from the patient perspective, the aspect of how hard the decision is to do. And so you still have the question whether you need to bring a device into the blood vessel. It's no comparison to drilling a hole and putting DBS electrodes in.
Although I've heard DBS described as minimally invasive as well in the past.
Yes, and I have been told that it looks far more terrible than it actually is, and that the looks might be one of the reasons why it's not.
Just one thing before we leave that. I mean, you've done sort of like forward modeling of electric signals and magnetic signals, and for electric signals, you want this electric head model that you can maybe get from MR studies, MR measurements. Can you also get sort of like an equivalent for the mechanical simulations from this MR from the same?
Very good for FUS, because what you need there primarily is the description of the heterogeneity of the skull. So you have these two cortical layers with the cancellus. The cancellus is very variable also between people, and that is the dominant factor in compensating for phase aberrations to get the focus. And for that, the Hounsfield units in CT are good, though it turns out that while Hounsfield units are usually considered rather universal, the scan parameter have a significant impact on the focality of FUS, and so there's a need to also work on standardizing that.
Of course, we as computational neuroscientists, we are sort of we are a priori fans of doing computational neuroscience to solve clinical problems. But you, Nada, you worked with medical doctors and clinicians in R&D. Are they as convinced about this as you are, or?
I think that depends, but I mean, my first postdoc supervisor was a neurophysiologist, so already towards a more kind of quantitative approach, I suppose. But he was really keen to do some modelling, or get somebody to do some modelling actually, and he got one of these discipline hopping awards, so really wanted to kind of bring things together. And so a lot of the early modelling that I did on deep brain stimulation was kind of driven by his curiosity about actually measuring local field potentials, which was his expertise, from deep brain stimulation electrodes, and what happens at the electrode brain interface, and how we can model that and understand that in terms of the physics, really, I suppose. And then as kind of time went on, so my PhD was about thalamocortical networks, and I'm still interested in doing those kind of more dynamical models as well. And so I've brought that into the work on deep brain stimulation as well, but using that as a different kind of approach to looking at how stimulation changes things from a maybe more mechanistic.
Because one of the reasons I'm having this, asking you, is also because there are obviously young people in the audience and young people listening to this podcast who may be sort of like considering to go into clinical neuroscience as a career choice. So, of course, one career possibility is sort of working in clinical groups. And would you recommend that, or what's your experience? Or?
Yeah, I think so, because obviously you get a different perspective, and you get exposure to going into DBS or whatever surgeries potentially, and you see things from a different point of view. And also, if you're working with data, I think it's great to actually see how data is collected, whether that's in clinic or in labs or whatever. That's always a good experience, I think.
I think also that the first applications for this technology tend to be for diseases where there are no other alternative treatments. So, for example, people who had lost their hearing due to the breakdown of the inner hair cells, there is no other treatment for them but a cochlear implant. And so, but it did take time for the cochlear implant to show that it was safe and effective, and now that's a very widely used technology. And I think that's been at the forefront of really demonstrating to doctors and patients that it is safe, and therefore these other technologies or other conditions can be considered. So, for example, with our retinal implant technology, that was targeted at retinitis pigmentosa, for which there is, or has been until recently, no other possible treatment. So I think we're seeing the same thing with brain-machine interfaces, that it's the people who really are quadriplegic or motor neuron disease for which there aren't any treatments, that they're the ones who are very keen to develop and be part of the journey to develop the technology.
Yeah, I guess especially with paralyzed people, like BrainGate was sort of like a company like 20 years ago, and now of course Neuralink is sort of getting a lot of attention. Whereas it seems quite impressive work that you could do this wireless, and it's like Elon Musk putting all this engineering into this. But we're going to say something, Esra?
Yes, I think maybe something important about what you said about the acceptance of modeling in the clinic. I mean, besides the fact that there's a generational aspect and that younger generations of clinicians expect modeling, there is also an aspect that maybe comes with responsibility. Modeling is it's very easy to produce colorful, impressive images. And you can impress with that, and you can impress the clinicians, and you have maybe also a responsibility of making sure that it's not suggesting a degree of knowledge about what happens inside the patient that is not really real. But that's a strong factor. And on the opposite side, you can do the best possible model, but if it's not respecting clinical constraints in terms of the degree of expert training, the time it takes from getting images until you are ready for the treatment, your expectation in terms of imaging that is done to give you your input data, if it's not respecting clinical constraints, it won't be accepted. It has to be something that can be done with a reasonable amount of resources. And if modeling can make the necessary compromises between the best possible model you can do and the one that is the best possible you can do within the constraints of the environment in which you have to operate, then it's a very welcome thing.
Because this getting improvements for like medical devices, I was involved in one neuroprosthesis project in the US, and then this FDA, this federal whatever, something that approves medical devices, they were like involved from the get-go almost, because it's really takes a lot of effort and persistence.
The regulators like the FDA have put out now an active demand to increase the role of modeling in regulatory approval. So nowadays they encourage to make use of in silico regulatory evidence, and in cases such as MR implant safety, they demand it. So modeling is also a methodology that is a way of demonstrating both the aspect of safety and efficacy. Those are the two keywords for regulators. And especially on the side of safety, modeling has become an important part of how you talk with regulators.
So, I mean, like another career path, in addition to working like in clinical, maybe at hospitals and so on, is actually working in a company or even starting a company. And I think you've been involved for a long time, Tony, with something called bionic vision, right?
That's right, yes.
And that's you've probably many challenges to overcome.
Indeed, and part of it was the regulatory environment. So we actually, I mean, this is actually part of what I learned from Graham Clark, was to actually get the regulators involved right from the very start, because that's absolutely critical to taking it through to being a commercial product. But also getting the clinicians really involved and doing focus groups with patients. It's really crucial to make sure that the technology is usable by the patients themselves and is also surgically implantable. So we had the surgeons involved in developing the surgery and developing the shape of the implant. All of this sort of thing really needed to be thought about right from the very start in order to make it viable as a commercial product.
Yeah, because I mean, then of course you need some kind of financial muscles, because I just read that there was like an American company trying to do the same as your company does, Second Sight, and they had some problems, right? I mean, I don't know what really happened to them, if they're still operating or...
That's right. In fact, they changed their device halfway through, so they actually went from having a retinal device to having a cortical implant, so going into the visual cortex.
Yeah.
And in fact, I think that they've now actually adapted that to other conditions, so I'm not sure that they're actually even doing anything in vision at this stage. So it is, all of these devices involve considerable risk. And so it does require an evaluation of what are the patient group that you're targeting, how many of the people actually have that sort of condition, and how many of them are willing to actually be the first adopters of the technology. That's why it's absolutely crucial to work with the patients and with the clinicians, because they really know what's sort of acceptable there. But it is difficult, there's no question about that. and there are significant hurdles. It's a very risky thing to do, and I think that was why having the cochlear implant as being the first one off the rank and actually getting that accepted has been so crucial for the development of the technology.
And I guess also it's the question of, I don't know how many people who are, if this retinal implants that your company makes, if that is approved and is working, I mean, how many, what's sort of like how many patients are we talking about?
So what we did with the project that we had was to do a pilot study with 4 patients, and it was actually only designed so that it would actually only be for a limited period of time, initially 12 months, but actually that got extended to 18 months. And then their device was turned off. It was actually, for most of them, it was left in with the option then that they could actually have the final product. The second trial was actually undertaken by the startup company itself, Bionic Vision Technologies, and they're just concluding that now with another cohort of patients. And they're using the data from that then to do a full-scale clinical trial that will be an international one that involves more patients and will feed into the FDA sort of regulatory processes.
So you are still optimistic?
So I think it's still challenging, but you know, like that's the nature of all of these devices.
I mean, when I tell people about this, that actually can sort of make sort of blind people see again, it's just like sounds too good to be true almost. So it's really fantastic.
But I think what we notice is that it's the patients who have the most severe loss. So with the cochlear implants, for example, it was patients who actually had no hearing whatsoever were the first ones. What's happened is that technology has become so successful that even patients with severe hearing loss who still have some hearing will preferentially get now a cochlear implant because it provides such good quality of living for them.
Esra, you have started several companies or have been involved in the starting of several companies.
Involved.
Yeah.
I really don't see myself as an entrepreneur. That's our boss who does that. But yes, we have been creating 3 big and about 5, 6 small ones along the way that co-finance our research. The first one was in the field of radiation and mobile phones, so outside the medical area. The second one then was when the first one was not interested in anything medtech or life sciences because that's too risky. So our simulation softwares and MR safety testing equipment was then commercialized by that second one. And then when we had a bit of a breakthrough with MIT, Harvard, and Imperial on non-invasive deep brain stimulation, there was a request from the research side to produce corresponding hardware, so we also created a little spin-off company. Along the way, goal was also to give the possibility to some of the younger researchers to be part of that experience of creating.
But you're not rich yet, or?
Sorry?
You're not rich yet, or?
No. I sometimes regret I didn't buy more of the stock, but no. But we are definitely involved, and we feel maybe another satisfaction of seeing that the work that we do ends up not just being a tool for our own research, but that it finds a broad acceptance. It maybe also forces us to think about the more general applicability of the things we develop. We need to turn them into building blocks for a wider application if we don't want to have them limited to the actual research that we do, but to be useful to a broad. And that way of thinking, of trying to identify components that are of a broader usability and how you can design them to be combinable and to be used for things that we might not know ourselves, that brings a mindset that's also nice.
But I think for me it sounds like it's a lower threshold for making a software company. You have like this Sim for Life, which is that some software to aid in this kind of explorations?
So we have 2 softwares that we developed. Sim for Life is this commercial one that was commercialized by ZMT Zurich MedTech.
So what does that software do?
It simulates what I said earlier, that you have physical exposure of anatomical environments giving rise to physiological or biological impact, and doing that also on the basis of image data. So it's used for everything from cancer therapy planning to neuroprosthetics that we mentioned earlier, or brain stimulation. And then we created a second software which is called OSpark, which is an open source software hosted on the web that has quite some success. We just had our 2000th person joining, and the goal there is to encourage people in the audience, for example, to take their research and make it such that it can be reused, that it can be discovered, that it runs in a sustainable and reproducible manner, and that it can be combined with other people's software. So what people can do with these modules is to create little services out of which pipelines can be generated. Those pipelines can be wrapped into a user interface and deployed for research clinical applications. And because we encapsulate runtime environment, the whole thing is in the cloud, it means that it becomes a bit of independent of your specific operating system. We kind of created it as an alternative to the vision of somebody we were closely working with that wanted everybody to go to a common language to express things a bit in the concept of systems biology. Everybody adopts the same language, formulates the things in a way where you have then energy, mass preservation, these kind of things. And we felt that people do not change their tools. People have established workflows for 10, 20 years in the lab. What we need to do is a way how people can keep working the way they always have and still produce something that fits with the work of others. And so with OSpark, funded by the NIH, we have created a platform that aims to make computational neurosciences reproducible, sustainable, and also shareable, publishable.
So we're getting closer to the end here, but there are all these new technologies that has been developed, maybe particularly AI, machine learning, and also, I mean, obviously molecular biology and technology, and I guess also brain imaging, but that's been around for some time. So what are sort of, how are these new technologies going to help to make good new, well, clinical treatments, including these like neurotechnology devices or whatever? Can anyone dare to say something there? Or
maybe I can comment on that, yes. So I have very ambivalent feelings about AI, particularly seeing that I've seen a lot of people replacing a biophysical understanding for the mechanisms by enough data. So if you have your big data, you can learn from it, and you might be able to have an effective therapy on that basis without necessarily understanding why it works. And on the one hand side, as a scientist, that hurts. On the other hand, as a patient, in the end, you want something that works. And if that works, the question is rather, does it allow you to make the next jump then that you can do because you understand the things? But then let's not forget that AI has a whole branch of AI that produces interpretable results. You have autoencoder strategies where you can identify lower dimensional representations of complex phenomena that become again completely interpretable. I think what's important is that we do not just solve the problems by saying, I don't need to understand them anymore, let's just get enough data to. I think that would stifle both our scientific purpose of trying to understand, but also in the longer term, the ability to innovate. AI can be used in a way where it also advances understanding.
Anyone? Nada?
Yeah, I was going to talk about, closed loop stimulation in DBS, which kind of overlaps a little bit with what you've just said in a way. So, I mean, I talked about this like volume of tissue activated as one approach of closing the loop, trying to kind of choose the clinical parameters that will maximize that. And in a way, that's also, overlooking maybe all of the intricacies of the mechanisms, because it's just one way of quantifying something. And it does correlate well with clinical outcome, but it might not tell us everything we want to know about DBS. And then the other approach to closing the loop in DBS is using simultaneously recording from the electrodes the local field potentials and then using that as your control signal to just switch on and off. And again, I suppose that kind of doesn't necessarily get at the heart of mechanisms, either of disease mechanisms or of the actual mechanisms of, stimulating the tissue. So, useful approaches and like you said, like these can still provide good, useful approaches that help, but I think there's still a lot to be done in terms of understanding mechanisms.
And I guess I wouldn't want to make myself a hostage to fortune by predicting what AI is or isn't going to do in the future. I think we're really at the very start of that development. I think there are enormous opportunities there, but I think there are also the caveats that people have mentioned. I think one of the things that's actually a little bit surprising is, we all fly in aeroplanes, and we were talking about this earlier. The model that we actually have of controlling an aeroplane is actually incredibly simple. Like, you don't need to actually solve all the Navier-Stokes equations about airflow and all this sort of thing in order to have a controller for even a large jumbo jet. The model can be relatively simple so long as you actually sort of understand how the model works and validate that. And I think this is what we're also discovering with the models of the brain, is that we perhaps don't need to have every detail of every synapse and the difference between all of the microstructures there. Having a relatively simple model that actually captures the key elements of it can actually be incredibly informative for these control purposes that we need for neuroprostheses and brain-machine interfaces.
The problem, I agree with you, and nothing against simple models. The problem is that AI might not be simple in its interpretation, and when it acts as a black box where it makes a prediction that you cannot verify itself, what you said earlier, you need to validate the suitability of a model within a given context of use. That's what the regulators will demand. And validation of a black box model and ensuring that you have the necessary safety margin is much harder than when you have an understanding for it. And so you will have, there are forms of models like physics-informed neural networks where you can in the end take the results, put it into what your assumptions were and confirm that your prediction is not an outlier. But there are many forms of AI where you have no way in the end of verifying that you are not in this, for this specific configuration failing. AIs, because of the nonlinearity and their form, they do not guarantee continuity and smoothness of response surfaces.
Absolutely, I agree completely. So I wasn't advocating the use of AI models for that by any means. So I guess the models that we actually use to understand the brain, even at the macro level, would be things like neural mass models. I do a lot of work myself on sort of spike-based models. So there is a gradation of models there that we can actually use. But I think your caveats about the use of AI is very correct.
For model order reduction of such a complex model. And in that way, first of all, you have transparency and you end up with something that you can again manipulate versus the extremely high-dimensional parameter space that you're using in a neural mass model.
Okay. So I think the final thing, I just heard outside when I just talked about this podcast, the topic, and one person said, well, isn't sort of like the neurotech the new cool thing? Is that, do you have that feeling that sort of that's like getting maybe interest from, I don't know, investors and stuff? Are they looking at neurotech as an interesting niche, you think?
You mentioned earlier about Elon Musk, and I think perhaps one of the few good things that he's done is actually, this interest in neurotechnology as being one of the areas that really will have a big impact upon health and for patients who actually have very severe conditions. So
but he sort of wants to have it so that he can connect to computers so that they're not out-competed by AI or something, right? So, but anyway, it's certainly for the patients who get this implant, it seems to be making their life much better. So.
That's right. We've been talking now about implants. Let's not forget that with aging as one of the big challenges of society, it's not necessarily always about implants. There is neurotech, also other forms of intervention that can play key roles in aspects such as cognitive or Alzheimer's and Parkinson's disease with increasing incidence. And so these are some of society's biggest challenges. And then in the field of neuroprosthetics and electroceuticals, we mentioned blood pressure earlier. Another very dominant factor in the question of the sustainability of our health system, where with bioelectronic medicine there are new approaches for fields that have maybe already explored what pharmaceutically is feasible at the moment.
Okay, I think this is a good place to stop. So also that I think that people could go to the poster session. So thanks a lot to the panel and thanks a lot to the audience.